cell division is a process of generating two identical cells from a mother cell. In bacteria, this happens by firstly the chromosome replication and segregation part, and the next step is the cytokinetic event, which is actually the division of the cytoplasm. In bacteria, this happens by a process called as binary fission. First, the cell will elongate. This is of E. coli or a rod-shaped bacteria. Once elongation happens, side by side septum formation will go on, where chromosome segregation will also be taking place, which means chromosomes will be separated into the two cells. Finally, the septum will be completed and the cells will separate by the action of hydrolase, an enzyme. Now, we will first look into an overview of the steps of cytokinesis. This in E. coli takes place by the formation of septum. Several of the proteins, some cytoplasmic and some integral proteins, are involved in the process. The cytoplasmic proteins will first assemble into a ring-like structure at the central position of the cell. Then this will call for the integral membrane proteins, which will also be recruited. This structure that is finally produced is known as the divisome. Now, after the formation of divisome, they will also help in the septum formation. That is actually the invagination of the cell membrane and the cell wall material. This invagination will continue to give a complete septum, and during the process, these proteins will be disassembled and will be moving into their respective locations within the cell. All this is happening and the cell is elongating in size. So, at the last stage, the septum is complete and already they have started moving apart. And finally, this will be cleaved by the hydrolase action and the two cells will separate. So, with this overview of cytokinesis, we move into more detailed account of these proteins that we have just come across. How exactly they help in the formation of a ring-like structure at the center and then followed by the septum formation. The first among these proteins, or the most important one, is the FTSZ. Now, this is a protein, cytoplasmic in origin, and is found in many different bacterial cells. This is the molecular structure of FTSZ. It has been worked on by several scientists. Now, this particular protein is a tubulin-like protein that is seen in eukaryotes, helping for this kind of cell division act. In prokaryotes, it is FTSZ. Now, this is the earliest acting protein in any cell division event.
Ma'am, is there any way to prove it?
Yes, several works have proved that FTSZ is the earliest acting division protein. Okay. Because if there is a block in any of the FTSZ production, then divisome will not happen. Secondly, if FTSZ is not recruited, other proteins will also be not recruited. That means FTSZ guides the recruitment of the other proteins. These have been shown by experiments. FTSZ is a very highly conserved protein. It is seen in many different bacteria, but there is an exception. L forms, which are cell wall less bacteria, they do not have FTSZ. Obviously, there is no cell wall, so cell division act will not happen in that fashion and FTSZ will not be seen there. Okay. Now, Bai and Leeuwenhuis were working with this FTSZ and they showed that this protein forms into a ring-like structure. Thus, they discovered the Z ring. These FTSZ molecules will arrange in head to tail fashion and will give rise to filaments. These filaments will now arrange properly so that a ring is produced at the mid region of the cell. The function of the Z ring is actually to constrict so that the cell division can go on. The invagination event of cell membrane and cell wall can take place only when there is a constriction by the Z ring. Other hypotheses are there that there are many more functions of Z ring, but this is the principal event which is there for cell division process. Now, moving on into this Z ring that we are discussing, can you see how exactly the polymerization of FTSZ has taken place and a ring-like structure is getting formed? This ring anchors to the cell membrane or cytoplasmic membrane. Now, that does not happen just like that. There are many other proteins which orchestrate this. One of it is ZIP-A. There is another one called as FTS-A. These two help in the anchoring of the FTS-Z ring onto the cytoplasmic membrane. Now, once this happens, the other proteins like FTS-I, FTS-K, FTS-W, not shown here, they start coming and joining this complex, finally to give rise to a structure called as the divisome. This is a typical divisome apparatus as seen in E. coli. Okay. Now, because the picture does not have all the proteins here, we have a list of all the proteins which can be functional in a division apparatus or a divisome. Now, you can see FTS-A, ZIP-A. Sometimes this ZIP-A is replaced by ZAP-A in many different cells. And the function of these are to anchor the Z ring to the plasma membrane. They hold the Z ring in position. Next comes FTS-E and FTS-X. Now, most of the function is not known, but it is postulated that they may be helping in some kind of a salt transport, which may be required in the cell during the division process. FTS-K is very important because it helps in chromosome segregation and separation of the chromosome dimers. So, this event should take place with the help of K so that the next steps can proceed. FTS-QLB. Now, these also the functions are not very well known, but postulated that probably they may help in the peptidoglycan synthesis. FTS-I and FTS-W are also involved for peptidoglycan synthesis. And then we have AMI-C and ENV-C, which are mostly for amidase and hydrolase activity respectively, meaning they will finally help in the cleaving of the dotted cells by the amidase and hydrolase activity. So, this gives us an account of most of the division proteins with their functions as seen in E. coli. Other cells may have some other proteins replaced in place of any one of these. Two systems are involved. One is a main system, the other is a nucleoid occlusion phenomena. The coordinated action of these two decides the location and timing of the FTS-Z ring formation. Now, we will be seeing the mean system in slight detail. Now, the three proteins mean C, D and E are involved. Mean C, D and E have been analyzed in E. coli. Similar proteins are present in other cells but may slightly differ. Let us see what is the action of these three proteins. Now, initially mean D bound to ATP will form polymers at the inner face of the cytoplasmic membrane. Only when mean D is bound to ATP can they polymerize in this fashion. And once the mean D is polymerized, mean C, another protein, can come and bind to this mean D, giving rise to a complex called as the mean CD complex. Now, there is the third protein called as the mean E. Mean E is a mean CD complex inhibitor. That means it inhibits the formation of mean CD complex. It also forms in the form of a small ring quite close to the pole. This is one pole. Now, when the CD is producing complex here, this starts producing a ring here. This ring catalyzes the conversion of mean D ATP to mean D ADP. So, the ADP now associated with mean D can no longer bind to the inner face of the cytoplasmic membrane, because that can be done only when ATP is bound to mean D. So, when mean D gets displaced, mean C will also be removed, because they generally remain in a complex and C is activated only by mean D ATP. That means when the mean Eve ring forms here, you can see they are moving on, catalyzing the depolymerization and removing these proteins into the cytoplasm, right? Now, as this happens from one pole, these proteins move to the other pole and in a similar fashion create a CD complex. Then mean E will also move to this pole and will make a similar activity. Now, that means what? The mean CD complex will oscillate from pole to pole. This takes place approximately 15 seconds in the case of the E. coli cells.
Ma'am, if there is mutation in any gene coding for mean CD, what will happen?
See, what will happen is the mean CD will not be able to prevent the Z ring formation at the cell pole, because mean C is the principal inhibitor of FTZ localization. And because mean CD is present at poles, FTZ formation at poles will not occur. So, if there is a mutation for CD, then they may not be occurring at the poles, and thus the poles will be free for FTZ ring formation. Okay. Now, you can understand if a FTZ ring forms at a polar end, then there will be a mini cell produced from this side and a large cell produced from this side. The large cell may contain all the components, but the mini cell may not have all the components. So, we will have something like an asymmetric cell division, and this mini cell may not be surviving in the population next. This is how we understand the speciation of FTZ localization. And the 15 seconds in E. coli, that is the time needed for the oscillation of mean CD from one pole to the other, also provides for a timing. When exactly should the FTZ form polymers at the center? Now, coming to the nucleoid occlusion phenomena here, the nucleoid is actually the chromosome of the bacterial cells. We call it nucleoid because they are not enclosed in a membrane bound organelle. Okay, now the nucleoid replication will start, and you can see here that the nucleoid is enlarging in size, which means the chromosome numbers are doubling, increasing. Now, after that, the DNA or the chromosome will segregate. That is, they are moving to the respective polar ends. Once the replication termination has happened, the two nucleoids you can see are already existing in the cell. Now, what is this nucleoid occlusion phenomena? When the nucleoid is present in the central position, they are associated with proteins called as NO proteins, nucleoid occlusion proteins. NOC, NOC and SLMA are two such nucleoid occlusion proteins associated with E. coli cell. When the NO proteins are present over the nucleoid, when the nucleoid is not replicating, the effect is known as NO effect, and no FTZ ring can be formed just over the nucleoid. The NO effect will prevent it. But once the cell has replicated its chromosome and segregated, this mid-cell area now has no NO effect. Okay. So, then the FTZ ring can form here. So, both the expression here, that is the mid-cell region being free of the nucleoids, as well as the mean CD only forming at the poles, leaving the central or mid-cell area free, helps us to understand that FTZ will localize in this mid-cell region. Okay. So, the timing and the location of FTZ correlated with the nucleoid occlusion and mean system can be understood by this slide. Now, after this, we move on to the other proteins which are occurring for the division process. In case of E. coli, we see that several proteins in the genera MREB are known for making or maintaining the shape. There are several such proteins like MREB, MREC, MRED, MBL. All of these in E. coli help in keeping the rod-like shape in the E. coli cell. There is a spiral of MREB proteins, as you can see here, produced. The FTZ is here, but these also form spiral rings, and these will help in the integrity and cell shape for the rod-shaped bacteria.
Ma'am, does all the bacteria possess this MREB protein?
MREB is seen in the rod-shaped bacteria. Now, what happens with the coccal cells? they do not have this MREB, and that is why their shape is a spherical one. They cannot retain a shape just like this. So most of the coccus, MREB proteins are not seen. Even MREB-like proteins are not seen. Now, as we are discussing the proteins for determining cell morphology, some more interesting facts comes with the next bacterium of choice, that is Caulobacter crescentus. Now, in Caulobacter crescentus, along with the FTZ protein, MREB proteins which form spirals, again you can see here, there is an additional protein called as crescentine. This crescentine, by maintaining the rod-like shape, is also helping in the maintaining of a curved appearance. So most curved bacteria like Vibrio, Caulobacter crescentus, these have additional proteins which will help maintain their curved morphology. This is crescentine in Caulobacter. Now the last stage in any cytokinetic event is peptidoglycan synthesis. The cell elongates, so new peptidoglycan material should be introduced. But we have a resynthesis event here. Now, as you can see here, this is a cell membrane. You will have a growing point of the cell. That means where new material should be incorporated in. Already existing peptidoglycan chains are seen and newer material has to be incorporated. That happens by the precursor molecule. A precursor molecule is a pentapeptide attached to N-acetylmuramic acid. N-acetylglucosamine then attaches to this muramic acid and pentapeptide, giving rise to a precursor molecule. Now, this precursor molecule attaches to a carrier known as bactoprenol. This bactoprenol is hydrophobic and it goes inside the cell membrane, transporting the precursor into the right place. Now, after it is placed, there are action of enzymes like transglycosylase. These will form the bonds between the existing NAG and the incoming NAM. A glycosidic bond will be formed by transglycosylase activity. The existing cell peptidoglycan needs to be broken down at places where new material has to be incorporated. So that is by autolysin activity. We move to the next slide where we see the last step of transpeptidation, meaning the cross link between the peptide bonds. Now, you can see the NAG and NAM here with the pentapeptide, alanine, glutamine, DAP, alanine again. So, the two adjacent peptides, peptidoglycan molecules will form the peptide bond here. Here you have a transpeptidation reaction and one of the alanine here is removed. So, this is the final step where you will see a bond, peptide bond is already produced here. This is principally in E. coli in a gram-negative cell. In a gram-positive cell, this will not be DAP, it will be L-lysine. That is the chemistry there. So, with this kind of a peptidoglycan synthesis, we come to almost understanding that this is how a cell will divide by the cytokinetic event. But we can also look into some more examples. Typically, we discussed in Caulobacter crescentus and we will discuss the Streptococcus pneumoniae. These two will have additional kinds of cell wall synthesis mechanisms. First, let us see what happens in Caulobacter crescentus. Now, division in Caulobacter crescentus takes the help of proteins like MIP-Z, PAR-B. FTZ is here. Now, Caulobacter cells have two kinds of cells. One is a stalked cell, you have the stalk here, and the other is a motile swarmer cell, which is not shown here because cell division event takes place in the stalked cell only and not in the swarmer cell. Now, initially, this is the origin. Origin was discussed in earlier lecture, the chromosome C point. Okay. Now, that will be associated with the PAR-B proteins. These will associate with the PAR-B sequence seen in the chromosome origin of Caulobacter crescentus. Once this happens, MIP-Z will now come and bind with PAR-B, to give a par B MIP-Z complex. Now, this will start to happen in only the stalked end, and later, once the chromosome segregation happens, the origin will now be replicated, because if chromosome replicates, origin will also be replicated. So, once they are getting segregated, the similar event which has taken place at the stalked end will take place at this end also. And by these will be pushed to the other end or other pole, the non-stalked pole. Now, as they are moving to the other pole, MIP-Z that you can see here is a FTZ inhibitor. So, as soon as these come, the FTZ will be forced to move from this end and come and lie at the center. So, this is a different kind of an event, as you have seen here. These are getting accumulated at this pole and the MIP-Z here, and the FTZ then moves into the center. This is not exactly a spherical bacteria, it is a ovoid kind of a cell. Now, here two machineries are involved for the last stage of cytokinesis, that is peptidoglycan synthesis. Okay. The two machineries are one for the peripheral PG synthesis and the other for the septal PG synthesis. What do you mean by peripheral PG synthesis? The peptidoglycan which will be synthesized at the peripheral or lateral regions, these portions. Okay. And the septal machinery is the one which will produce the septa. Now, in most bacterial cells, as we have seen earlier also, in E. coli, it is just a one kind of a machinery that produces the septal PG peptidoglycan. But in case of the Streptococcus species here, we see the two are involved. One continuously carries on the septal PG synthesis, the other the peripheral PG synthesis. Although we have heard of Streptococcus as principally cocci, they are actually ovococcus, coccal cells which are slightly oval. And the two machineries that you will be seeing here are actually operating into two diverse reactions. For simplicity's sake, in the diagram they have mentioned here as two separate systems or machines, but they are all the one. One produces at the peripheral region, the other at the septum region. Now, this one is actually the PG hydrolytic machinery which acts for hydrolase enzyme. Once the peptidoglycan will be produced, newer one, later these cells needs to be separated. So, there should be some hydrolase action which will cleave the peptides and the peptidoglycan synthesized, which are joined now into two separate daughter cells. Now, along with this, we also move into the second part here. This gives us all the proteins which are required. In this left side, you can see the septal machinery, means the proteins which are required for the septal PG synthesis. And in the right here, you can see the machines or proteins required for peripheral PG synthesis. Now, let us consider the proteins here. PBPS stands for penicillin binding proteins. Now, these are principal proteins seen in the cells of most of the bacterial rod-shaped cells. We are seeing here in case of even the coccal cells. Now, PBPs are of several types. PBP1A, GPSB, these two are others kinds of PBPs are the common factor that you see in the septal machine and in the peripheral machine. Now, PBP2X is not here but replaced by PBP2B. Likewise, FTSW is present here in case of the septal machinery, while rod A is present here in peripheral machinery. MRED, MREC, these are not present anywhere in the septal machinery, but you see here in the peripheral machinery. So, I already mentioned about MREC and D, which may be involved in ovococcal cells. But if you remember, we just mentioned in our earlier slides that MREC and D or MREB, these are not present in the coccal cells. Do you remember?
Yes, ma'am.
But this is an exception to it. Now, here what happens, the EZRA along with GPSB is very nicely anchored and associated to the FTS divisome, the divisome proteins, which will orchestrate the peptidoglycan synthesis along with the division of the cell. While in case of the peripheral machinery, this attachment is not seen. This does not happen. Like there is no EZRA here, and thus this kind of a attachment is not seen. So this rightly explains that several proteins we will come across in some special cells. Different proteins will be present. You cannot go for a generalized structure like that. And that is why we take E. coli as the model organism and study what is happening there. What are the protein components there? That is what we have done here, and we have kept 2 such different slides wherein you can have examples of some other related proteins. To conclude, we may state that the cell dividing by binary fission in E. coli will culminate by the cytokinetic event. But during all this process, the DNA replication, its segregation into the daughter cells will all be taking place. The proteins for the divisome, at assembly of the divisome, FTS Z ring formation, all this will proceed along with the main proteins, the nucleoid occlusion phenomena, which will provide for temporal and spatial specificity to the cells, will help us decide when exactly and where exactly division will take place. But the cytokinetic event is not complete until the last step, that is of peptidoglycan resynthesis, which we have seen in detail, and at the last stage hydrolases will cleave the arranged or septal cells into finally 2 daughter identical cells.